All right. In this explainer, we are diving headfirst into a real-life scientific detective story. I mean, this was a mystery that took over half a century to crack. It's the story of a killer that was so elusive, well, ghostly that it slipped through every single trap that science could throw at it. And in the end, it forced us to completely rethink the very rules of life itself. So how do you catch a killer that's smaller than light itself? I mean, that was the central challenge, right? You have to remember, back in the late 19th century, scientists were feeling pretty good about themselves. They were confident they had basically cracked the code of disease. But what they were about to do was start hunting for a true phantom, a culprit so impossibly small it literally lived in the shadows, way beyond the reach of their most powerful tools. Okay, so our story actually begins not in some fancy lab or hospital, but out in the tobacco fields of the Netherlands. Now, we're talking about the 1880s here, which was basically the golden age of the germ theory. You had giants like Pasteur and Koch proving that these tiny little things called bacteria cause diseases. The rules seem super simple, right? Find the germ, see the germ, kill the germ. Easy. Except, a really strange blight was just tearing through Europe's most valuable crop, and it followed none of those rules, not one. So you see this stuff on the leaves? Farmers started calling it mosaic disease because of this sickly mottled pattern it painted all over them right before they just withered up and died. And look, the economic stakes here were massive. So in 1879, a German chemist, a guy named Adolf gets called in to investigate. You could say he was the first detective on what would turn out to be a very, very strange case. So what did Mayer do? He started with this brilliantly simple experiment. First, he just crushes up the diseased leaves to get all that green sap out. Then, he takes that sap and injects it into a perfectly healthy plant. And what happens? Well, he waits, and sure enough, within about 10 days, those healthy plants get the exact same disease. Boom. That was the first crucial clue. He'd just proven the disease was infectious, and that sap? The sap was the culprit, the vector. But here's where the mystery really begins. Mayer, feeling pretty confident, puts the infectious sap under his most powerful microscope. He's totally expecting to see it just swarming with bacteria, right? But there was nothing. Zero. So he thinks, okay, maybe I just need to grow it. He tries to culture the microbe in a petri dish, and again, nothing. So he's got this puzzle. He knows it's contagious, but he can't find the bug anywhere. He eventually concludes it must be some bizarre, super tiny bacterium that his tools just couldn't see. He was so close. But the truth? Oh, the truth was way stranger. All right, so to solve this mystery of the invisible germ, science clearly needed a better trap. And as luck would have it, a brand new piece of tech had just hit the scene. It was designed to be the ultimate, foolproof bacterial sieve. This thing was supposed to end the debate once and for all. But instead, it just made the mystery even deeper. And this was the Chamberlain filter. It was actually invented by one of Louis Pasteur's own assistants. So you know it had a pedigree. The best way to think of it is like a water filter, but on absolute steroids. It was this tube of unglazed porcelain with pores so incredibly microscopic that while liquid could get through, no bacteria, and I mean no known living organism, could possibly make it to the other side. The liquid that came out was, for all intents and purposes, guaranteed to be sterile. So in 1892, a Russian botanist named Dmitri Ivanovsky decides to use this new gadget to settle the matter. His logic was perfectly sound. He thought, okay, if a tiny bacterium is causing this disease, this filter will trap it, no question. The sap that comes out the other end will be totally harmless. So that's what he did. He ground up the leaves, forced the sap through the porcelain, and collected this beautiful, crystal clear liquid that dripped out. And then he takes this supposedly sterile fluid and he rubs it onto some healthy tobacco plants. And to his absolute astonishment, they got sick. The thing causing the disease, whatever it was, had passed right through the bacteria-proof filter. I mean, this result should have been impossible. It flew in the face of everything they thought they knew, a direct contradiction of the germ theory. So this raises a huge question, right? What in the world could pass through a bacteria-proof filter? This should have changed everything. But, Ivanovsky was a man of his time. He had the perfect data right in front of him, but he just couldn't make that conceptual leap. His mind couldn't go there. Instead, he concluded his filter must have been cracked, or maybe the bacteria were releasing some kind of tiny chemical toxin that was small enough to slip through. He had the phantom cornered, but he just couldn't bring himself to believe what he was seeing. It was going to take another scientist, a Dutch microbiologist named Martinus Beijerinck, to come along a few years later. He would look at the exact same evidence Ivanovsky had, but come to a conclusion so radical, so out there, that it would completely shatter the old paradigm and give birth to an entirely new field of science. So, here's how Beijerinck brilliantly reasoned his way to the truth. He asked a simple question. Is this mysterious agent a chemical toxin, or is it a living organism? See, a toxin, a non-living poison, gets weaker every time you dilute it. It spreads out. But Beijerinck showed that he could take the infectious sap from one plant, infect another, then take the sap from that second plant and infect a third, and so on and so on. The disease never lost its punch. It was just as strong in the 10th plant as it was in the first. That could only mean one thing. The agent was multiplying. It was reproducing. It had to be alive. So in 1898, Beijerinck makes this incredibly bold declaration. He says, look, this pathogen is not a cell. It's not a bacterium. It is something entirely new. He gave it this amazing name, contagium vivum fluidum, which is just Latin for contagious living fluid. He was proposing a new form of life, one that had no cells, one you couldn't grow in a dish, but that could somehow reproduce itself once it got inside a host. And to give this strange new entity a name, Beijerinck reached way back to an ancient Latin word, a word that meant venom or poisonous secretion. He called his invisible, filterable, self-replicating agent a virus. And just like that, the field of virology was born. But wait, the mystery was far from over. Okay, so they had a name, virus. But what was it physically? Was it really a fluid like Beijerinck thought? Or was it some kind of tiny particle? The next huge breakthrough came from an American biochemist, a guy named Wendell Stanley, who decided to attack this problem from a totally different angle, not as a biologist, but as a chemist. Okay, so Stanley's whole approach was built on this one fundamental rule of chemistry. You can crystallize pure chemicals, like salt or sugar, into these perfect, beautiful geometric shapes. But what you cannot do is crystallize a living thing, a cell. A bacterium, it's just too complex, too messy, alive. So Stanley's big goal, his challenge, was to try and crystallize the tobacco mosaic virus. If he could pull it off, it would prove it was just a chemical after all. And in 1935, after what I can only describe as a monumental effort, we're talking about grinding up tons of infected leaves and using gallons and gallons of harsh chemicals to purify the sap, he did it. Stanley actually succeeded. He isolated these fine, shimmering, needle-like crystals from the infectious liquid. And this is where things get truly mind-bending. Because on his lab bench, these crystals were just inert chemicals. That's it. They didn't eat, they didn't breathe, they didn't move, they were dead. But, and this is a huge but, when Stanley dissolved just a tiny pinch of these dead crystals in water and wiped it on a healthy leaf, the plant got violently sick. It was unbelievable. He had proven that the virus exists on this weird, ghostly boundary between chemistry and biology. Okay, so let's recap. By the late 1930s, scientists knew a virus existed. They knew it could pass through filters. They knew it was some kind of chemical that acted like it was alive. But get this, no one in all of human history had ever actually seen one. And the reason was simple but profound. They were literally smaller than the wavelengths of light. You can't see something if light just passes right over it. To finally see this phantom, they were going to need a whole new kind of light. And that new light came in the form of the electron microscope. Now, by using a beam of electrons instead of light photons, this absolutely incredible invention could magnify objects to a level that was just unimaginable before. It finally gave humanity the eyes to see the invisible. And you really have to appreciate that this wasn't just one person's discovery. Not at all. It was more like a scientific relay race that spanned over 50 years and crossed multiple countries. Each scientist handed the baton to the next. Mayer proved it's contagious. Pass. Ivanovsky proved it's filterable. Pass. Beijerinck gave it a name. Pass. Stanley proved it's a chemical. Pass. And then finally, in 1939, a team in Germany took the final lab. So when they finally developed that first photographic plate, the ghost had a face. And it wasn't what anyone expected. It wasn't a fluid, it wasn't some tiny, misshapen cell. It was a collection of these perfectly straight, rigid, rod-shaped particles. For the very first time, humanity could see the face of a virus. And it was stark, simple, and utterly alien, unlike anything we had ever seen before. And from that very first glimpse of a single, humble plant pathogen, we have come to understand that we live on a viral planet. It's estimated there are 10 to the 31st power individual viruses on Earth. Okay, what does that mean? That's a 1 with 31 zeros after it. The number is so vast, it's practically meaningless to our brains. It's far, far more than all the stars in the known universe. They are, without a doubt, the most abundant biological entities we know of. And all of that brings us to the final and maybe the most profound question this whole journey leads us with. Think about it. The tobacco mosaic virus can sit in a crystal jar on a shelf as dead as a rock. But the moment it gets inside a living cell, it becomes this unstoppable replicating force. So this discovery, it didn't just reveal a new pathogen. It forced us to confront one of the deepest questions of all. Where exactly is the line between life and non-life? It's a question we are still grappling with to this very day. Thanks for watching.
Please like and subscribe.